Thank you very much. It is my pleasure and privilege to present to you some of our results from a study in Zurich. So I'm going to talk about white matter asymmetries in the developing brain, especially in the newborn brain. But before we get started, let me introduce you to some of the biological concepts of morphological asymmetry in very general terms. To introduce you to this topic, I have brought this illustration, which is the population level histogram of the laterality index of any morphological trait that has left or right side, so bilaterality. And in these plots, you see that we distinguish three different concepts. The first one is the so-called fluctuating asymmetry, which refers to developmental instability, which means actually a random departure from the mirror symmetry to either left or right side. And it's a phenotype, and it's not genetically regulated. It comes during development, so it's a variability. Second, the directional asymmetry, which is the non-random departure to any of these sides. And as you see in these plots, it's a genotype. It's determined by certain genes regulating development. And also, on top of that, you see that the fluctuating asymmetry contributes to the directional asymmetry that we see in these traits. Anti-asymmetry is also a directional asymmetry, but it doesn't have a preferential side. It's either left or right-sided. So talking about the brain, interhemispheric asymmetry and specialization of the brain is a fundamental property of many vertebrate species, including humans as well. And it's not a particularly new thing to know. We have known that since a century, since the pioneering works of Broca or Desjardins. So you see this illustration of destructive brain lesion. And brain asymmetries form the possible morphological basis for functional specialization. We all know this. And what we also found, or what the literature says, that this is not really a human-specific trait in general, although there are some of them which appear to be human-specific. Talking about brain development, why is it interesting to study asymmetry during brain development? If we study prenatal or early postnatal neonatal brains, we realize that the asymmetries that we see are the ones which are genetically determined and which are not modified by later life experiences or neuroplasticity. And of course, there's a variability, developmental instability in the prenatal brain as well. And also some pathologies might interfere with that, but we don't know that. The newborn brain appears to be asymmetric, and actually studies from the 1970s already demonstrated the planum temporale, which is thought to be associated with language, is asymmetric in the newborn human brain. So there seems to be a strong genetic determinant of brain asymmetry. And also in utero MRI studies reveal that the superior temporal sulcus is right dominant. So all these regions appear to be quite asymmetric early on during development. The purpose of our study was to use in vivo diffusion MRI in a multi-centric cohort of newborns to evaluate whether interhemispheric asymmetry exists in white matter structures, particularly directional left-right asymmetry, fluctuating asymmetry, the variability, and also anti-asymmetry was evaluated in our study. This is a multi-centric study, so we took a couple of studies publicly available and the data that is available in Zurich. So it's actually a analysis of 4 different data sets. 141 fetuses have been analyzed. Half of the samples were healthy developing fetuses, and as you see in this table, half of them are CHD or preterm, but 4 groups were formed and 4 variables controlled for the presence of pathologies and the different study groups in the statistical models. They were imaged at term equivalent age on 3 different imagers, which were also controlled in the statistical model. So let me just shortly summarize the image processing steps. This is pretty standard. First, we created a fractional anisotropy template by using the Albert Atlas, which is a T2-weighted neonatal atlas to which we co-registered the FE images. And this served as a reference to study asymmetry. Then we did DTI pre-processing on this neonatal statistical data, motion correction, eddy correction, and estimation of tensors by the restore model was performed. We calculated for this study the fractional anisotropy and something different, the anatomical connectivity map, which is actually derived from whole brain probabilistic cryptography. So the probabilistic index of connectivity for each pixel was stored in an image. This looks pretty similar to the FA, but it's actually something different. How we measured asymmetry, so we split these brains after standardization up to left and right hemispheres. The right hemispheres were flipped to the left, and then they were again co-registered with each other to make sure that there are no anatomical bias and they overlap, so the fiber tracks overlap with each other. And after that, we also did a tract-based spatial statistics, or TBSS, to make sure that actually we're statistically analyzing anatomically corresponding points in space in the same white matter tract. The statistical test is quite tricky when doing multi-centric analysis. So we did a paired design in which we controlled the left and right hemispheres of the same subject across the groups with each other. We controlled for age, so gestational age at the time of imaging, gender, presence of pathology, and the number of the study center which the newborn was imaged at. And also we controlled for the multicentric study by exchangeability blocks in the randomized program. And as you see here, we use the standard FSL-TBSS type of analysis to visualize the results. Second, we measured the fluctuating asymmetry, which is, as I said, just a variability metric of the laterality index, or the asymmetry. The trick here is that because it's a multicentric design, we have to normalize within group. So there's a normalization group within subject, but also within groups. And what we could calculate here is like the top 5 percentile values of fluctuating asymmetry. And third, we calculated the anti-asymmetry, which statistically it was possible to do a test which tested bimodal distributions across the population, which was done by the Hertig-and-Dip statistic. Let me show you the results. The directional asymmetry of white matter bundles, FA values, confirmed what is known from the literature, that the corticospinal tract, the posterior limb of the internal capsule, is left dominant already at birth. And already early development, you see these yellow to red spots there. And on the left, on the other hemisphere, we just depicted the right dominant fibers, which is the inferior fronto-occipital fasciculus. So these were the fibers which are reproducibly seen to be asymmetric across all study centers. But as you see in the distribution of the laterality indices, it's quite surprising that the actual laterality is really low, so it's 2-3%. So it's not very asymmetric. The anatomical connectivity maps showed a similar distribution for the corticospinal tract, sensory motor tracts, posterior limb of the internal capsule. Surprisingly, there was a difference, so it was the external capsule and probably the anterior part of the inferior fronto-occipital fasciculus, which was found to be right dominant in the ACM maps. Let's have a look at the variability of the laterality index in newborns. I depicted the top 5 percentile in red and the low 5 percentile of variability in blue for the FA and ACM maps. And as you see in these pictures, we found that high variability is the frontal, prefrontal regions, external capsule, and also parietal regions near the IPS, while low variability is seen in the corpus callosum and corticospinal tract. FA and ACM showed similar patterns. And regarding bimodal distribution, we did found similar significant pixels, but they were not overlapping across groups. So we concluded that there is no anti-asymmetry in newborns in the human brain in FA and ACM values. To sum up, sensory motor tracts such as internal capsule, posterior limb have lower fluctuating asymmetry. They are left symmetric. Leftwards directional asymmetry appears to be genetically coded in newborns before right-handedness in quotation mark can be learned. we confirmed rightwards asymmetry of IFOF. And what we were able to do, basically, it's a replication study of many other studies that performed DTI symmetry in newborns or during development. I believe our study is a bit more powered, but what is surprising that we have shown fewer tracts to be reproducibly asymmetric, and we have shown that actually in newborns the laterality index is much lower than in adults. So we believe that this initial genetically coded asymmetry may get more pronounced during later life by neuroplasticity and experiences. For the fluctuating asymmetry, frontal prefrontal tracts show higher fluctuating asymmetry, and let me just speculate here that, going back to the biological notion, that high fluctuating asymmetry means developmental instability. So probably these are more novel regions, evolutionary novel regions, which are thought to have larger developmental instability, and probably this is what we see being reflected in the higher fluctuating asymmetry of the white matter tracts. So thank you very much for your attention.
Thank you.
I'm looking forward to the questions.
Thank you.